Hi dear students. So in this session, let's see about the size, shape and arrangement of bacterial cells. Bacteria are prokaryotic unicellular microorganisms which lack chlorophyll pigments. Cell structure is similar than that of other organisms as there is no nucleus or membrane-bound organelles. Due to the presence of a rigid cell wall, bacteria maintain a definite shape, though they vary as shape, size and structure. When viewed under the light microscope, most bacteria appear in variations of three major shapes: the rod or bacillus, the sphere or coccus, and the spiral type, that is the vibrio. In fact, structure of bacteria has two aspects, that is arrangement and shape. So far as the arrangement is concerned, it may be paired like Diplococcus, and it can be grape-like clusters as in the case of Staphylococcus, or chains in the case of Streptococcus, and in shape they may be principally be rods, that is bacilli, spheres, that is cocci, and spirals, that is spirillum. Coccus. The plural form of coccus is cocci. It can be used to describe any bacterium that has a spherical, ovoid or generally round shape. It is one of the three distinct bacterial shapes. The next one is a bacilli, gram-positive rod-shaped bacillus bacteria and a member of the phylum Firmicutes. Bacillus species can be obligate aerobes, that is that they require oxygen strictly for their growth, or facultative anaerobes, that is having the ability to be aerobic or anaerobic. The next one is Spirilla. Spirillum is a genus of gram-negative bacteria within the family Spirillacea. The most common known two species in Spirillum is Spirillum vinogradski and Spirillum volitens. Let's see the size of bacterial cell. The average diameter of spherical bacteria is 0.5 to 2.0 micrometer. For rod-shaped or filamentous bacteria, the length is 1 to 10 micrometer and diameter is 0.25 to 1 micrometer. E. coli, a bacillus of about average size is 1.1 to 1.5 micrometer wide by 2 to 6 micrometer long. Spirochetes occasionally reach 500 micrometer in length, and the Cyanobacterium oscillatoria is about 7 micrometer in diameter. The unit of measurement used in bacteriology is the micron or micrometer, which is 1,000 of a millimeter. Bacteria in general is one-tenth the size of the eukaryotic cell. On average, the size of bacteria ranges from 0.5 to 5 micrometer. However, they can be as tiny as 0.3 micrometer and as large as 0.7 millimeter. The limit of resolution with the unaided eye is about 200 microns, and as many bacteria are smaller than this size, they are not visible with naked eyes. The bacterium Epiloecium fischelsoni can be seen with the naked eye. It's 600 micrometer long and 800 micrometer in diameter. One group of bacteria called the Mycoplasmas have individuals with size much smaller than these dimensions. They measure about 0.25 microns and are the smallest cells known so far. They were formerly known as Pleuropneumonia-like organisms, that is PPLO. Mycoplasma gallisepticum with a size of approximately 200 to 300 nanometer are thought to be the world's smallest bacteria. Thiomargarita namibiensis is the world's largest bacteria. It is a gram-negative proteobacterium found in the ocean sediments of the coast of Namibia. Usually it is 0.1 to 0.3 millimeter across, but bigger cells have been observed up to 0.75 millimeter. Let's see about the shape of bacterial cell in detail. Most of the bacteria have a rigid cell wall that provides a definite shape to the bacteria while protecting the internal components. Even though this characteristic is varied for the majority of bacteria, they vary in shape that allows them to be classified into different groups based on their forms. This wide variety of shape is determined by the bacterial cell wall and cytoskeleton. Bacteria with different shapes present different physical features to the outside world, and these features help cells cope up with and adapt to external conditions. It has been observed that bacterial shape contributes a measure of survival value in the face of nutrient acquisition, cell division, predators, attachment to surfaces, passive dispersal, active motility, and internal or external differentiation. As mentioned earlier, the three basic bacterial shapes are coccus, bacillus, and spiral. Cocci are round cells, sometimes slightly flattened when they are adjacent to one another. Bacilli are rod-shaped bacteria. Spirilla are curved bacteria which can range from a gently curved shape to a corkscrew-like spiral. Many spirilla are rigid and capable of movement. A special group of spirilla known as spirochetes are long, slender, and flexible. Arrangement of cocci. Cocci bacteria can exist singly, in pairs, in groups of 4, in chains, in clusters, or in cubes consisting of 8 cells. The cocci may be oval, elongated, or flattened on one side. Cocci may remain attached after cell division. These group characteristics are often used to help identify certain cocci. In this image, you can see the single cell coccus, the diplococci, the tetrad, that is the group of 4 cocci, and the cube-like shape, and the example is Sarcina, and the chain-like morphology of the cocci occurs in streptococci, and the grape-like cluster of cocci occurs in staphylococci. The examples of diplococci include Streptococcus pneumoniae, Moraxella casualis, Neisseria gonorrhoeae. The streptococci are arranged in chains as the cells divide in one plane. In tetrads, the cocci are arranged in packets of 4 cells as the cells divide in 2 planes. The examples include Aerococcus, Periococcus, Tetragenococcus. In Sarcinae, the cocci are arranged in cuboidal manner as the cells are formed by regular cell division in 3 planes. Cocci that divide in 3 planes and remain in groups cube-like groups of 8. The examples include Sarcina ventriculi and Sarcina ureae. In Staphylococcus aureus, the cocci are arranged in grape-like clusters formed by irregular cell division in 3 planes. Let's see the arrangement of bacilli. The first one is diplobacilli. Most bacilli appear as single rods. Diplobacilli appear in pairs after division. The examples of single rods are Bacillus ceres, and examples of diplobacilli are Coxiella burnetii, Moraxella bovis, Klebsiella, Rhinoscleromatis. The next one is Streptobacilli. Here, the bacilli are arranged in chains as the cells divide in one plane. Example, Streptobacillus moniliformis. The next one is the Coccobacilli. The Coccobacilli are so short and stumpy that they appear ovoid. They look like coccus and bacillus. The examples are Haemophilus influenzae, Gardnerella vaginalis, Chlamydia trachomatis. Next arrangement of bacilli is palisades. The bacilli bend at the points of division following the cell division, resulting in a palisade arrangement resembling a picket fence and angular patterns that look like Chinese letters. Example of palisade organism is Conibacterium diphtheriae. Next one is the arrangement of spiral bacteria. Spirula or spirillum for a single cell are curved bacterium which can range from a gently curved shape to a corkscrew-like spiral. Many spirula are rigid and capable of movement. A special group of spirula known as spirochetes are long, slender, and flexible. In this, the first one is Vibrio. They are comma-shaped bacteria with less than one complete turn or twist in the cell. An example of this Vibrio is that Vibrio cholera. The second one is Spirilla. The spirilla have rigid spiral structure. Spirillum with many turns can superficially resemble spirochetes. They do not have outer sheath and endoflagella but have typical bacterial flagella. Example is Campylobacter jejuni, Helicobacter pyrrole, Spirillum vinogradski. The third form of spirilla is spirochete. It have a helical shape and a flexible bodies. Spirochetes move by means of axial filaments which look like flagella contained beneath a flexible external sheath but lack typical bacterial flagella. Examples of spirochetes are Leptospira species, Treponema pallidum, Borrelia recurrentis. Other shapes and arrangements of bacteria include the filamentous bacteria. They are very long, thin filament-shaped bacteria. Some of them form branching filaments resulting in a network of filaments called mycelium. For example, Candidatus savagella. An example of the star-shaped bacteria is Stella. And the example of rectangular bacteria is Halarcula species. And the other one is the pleomorphic bacteria. These bacteria do not have any characteristic shape unlike all others which we have described above. They can change their shape. In pure cultures, they can be observed to have different shapes. Examples of bacteria exhibiting pleomorphism is Mycoplasma pneumonia and Mycoplasma genitalium. Another example of pleomorphic bacteria is Arthrobacter. And in this image you can see the overall bacterial shapes and arrangements of Coccus, Bacillus and Spirillum. The stalk bacteria or prosecate bacteria are a group of bacteria that create thin extensions of the cell wall from the main cell body. Many stalk bacteria are found in the alpha Proteobacteria including Caulobacter, Brevundimonas, Asticolis and Hyphomonas genera. As an unusual morphological event, stalks also are a target of study for trying to understand the fundamental biology of cell shape, that is, how and why bacterial cells alter their shape. Actinomycetes. Actinomycetes are unicellular gram-positive bacteria that belong to the order Actinomycetes. Members of this group are widely distributed in nature and can be found in a variety of habitats across the world. They have been shown to share a number of characteristics with fungi, for example, the mycelial growth. Actinomycetes can range from harmless bacteria to pathogens with resistance to antibiotics. The important genres include Actinomyces, Nocardia, Actinomadura, and Streptomyces. The actinomycetes are a diverse group. They are gram-positive, non-motile, non-sporing, and non-capsulated. They are straight, curved, or pleomorphic. They are arranged in chains or branching filaments, and they are related to mycobacteria and corynebacteria. Mycelium, the total mass of hyphae, is also found in nature. Actinomycetes are unicellular like bacteria but produce mycelium which is non-septate and more slender. Like bacteria, they don't have distinct cell wall and they don't contain chitin and cellulose. They produce hyphae and conidia like fungi. Certain actinomycetes whose hyphae undergo segmentation resemble bacteria both morphologically and physiologically. So the actinomycetes resembles morphologically to fungi and the cellular organization is typical to bacteria. Here you can see the morphology of actinomycetes. And this is how the actinomycetes look in the culture media.